Hello, dear students. I hope you all are fine and doing well. I welcome you all on behalf of Sri Jendra Rank John classes. Dear students, as you know, we have started with the our new chapter, that is the biological classification, and in a series, this is lecture number 2. So dear students, already in previous class we have discussed, we have understood that what are the different classification system evolved at a certain period of time, and it was given by a different scientist. Now, after the particular classification systems which we have discussed, that is two kingdom, three kingdom, four kingdom, five kingdom, six kingdom, and three domain classification. We have ultimately understood there are major five kingdoms are that, like kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Animalia, and kingdom Plant. In this chapter, we are going to deal with three out of this. We are starting today in this lecture with the kingdom Monera. In today's lecture, we will be dealing with the Monera in depth, in detail. You know, Monerans are bacteria. So, we are going to deal with this in detail. Okay. So, in this Monera, we are going to study bacteria in detail. Okay, dear students. So, let's start. So, if we talk in Monera, in kingdom Monera, we will be going to talk about the different organism. Like we will be going to talk about eubacteria. In eubacteria, we will study the different. First, we will talk about the structural aspect. Then we will talk about functional aspects. Functional aspects. Then we will going to talk about study of blue-green algae or cyanobacteria in detail. Then we will talk about mycoplasma. Then actinomycetes. After that, we will be going to talk about archaebacteria. So this many things we are going to study in this chapter, okay, in depth. So you have to go with me in a proper manner so you can understand this in detail. So first, we will be starting with the eubacteria, okay, and in this eubacteria, we will study in detail. This eubacteria, if we call them, they are known as true bacteria. They are known as true bacteria. Now the first thing, this eubacteria include a true bacteria and it include true bacteria. Now this bacteria are most abundant microorganism because if you take a one spoon of sand only, it have a millions of bacteria. So dear students, it is considered as the most abundant microorganism on this earth. Then why? Because they almost occur everywhere. We can say bacteria are cosmopolitan. Why? Because bacteria can be found everywhere, like on a soil, like on a stem or a plant, on animals, in a water. Everywhere you can see the presence of this particular bacteria. So they are found almost everywhere. Now, bacteria live in a different environmental conditions. Okay, they live in a different environmental condition. Sometimes you found this bacteria in a normal our nearby. Usually we see the bacteria in our near condition, but sometimes this bacteria can be found in some of the extreme conditions. You can see this treatment that they are found in some of the extreme habitats. Extreme habitats like hot springs are there. You can see the bacteria presence of it. Deserts also bacteria can be present there. Snow and deep oceans. There also there is can be a possibility of presence of bacteria, because human being cannot live in this different condition, or any other animals cannot live, but bacteria can live. That is a specialty features of this bacteria. Okay, so it is the thing, but it is important that very few forms can particularly survive there. So this is the one general structural aspect which is given here for the particularly you can say this bacteria. You can see here that this particular bacteria is having a DNA. Then this bacteria has its own cytoplasm, cell membrane. Can have a flagella. Okay, and 3 micrometer size is given. But we will be studying about the structure in detail. Okay, students. So we understood here that bacteria can be found anywhere. This statement is explaining the nutritional aspect that many of bacteria live in other organisms and parasite, and some of them are symbiotic. We will be dealing about this statement is given, but we will be dealing here. See here one example of some bacteria can be found in free living form. Some bacteria can be found in a parasitic form. Some bacteria can be found in the symbiotic form. So the example here given, that example is of a legume. In leguminous plant, some bacteria are there which live in a symbiotic association with the roots of this particular, we can say a leguminous plant. So what these red colored dots are the nothing but what a particularly root nodules which are found in the roots of leguminous plants. And these root nodules carry out biological nitrogen fixation by fixing nitrogen into the ammonia. Okay. So these nodules have certain bacteria. This carry out this type of mechanism, and they are nothing but which one? They are symbionts. Okay. So this way, bacteria can also help in the nitrogen fixation. Some bacteria can carry out, particularly we can say decomposition as well, and some bacteria can be like as a parasite. So we will be talking about that in a nutrition in detail part. Okay. So if you see here, okay, here you can see that bacteria are grouped into 4 categories based on their shape. Okay, so bacteria have a variety of shape. So let's talk about the particularly shape of bacteria. Dear students, because bacteria are found everywhere, so based on the environmental condition, there can be a different shape as well. So if we talk about shape of particular bacteria, you come to they know that the first important shape they may have, that is spherical. Okay, you can see this diagram. This is a particularly spherical shape bacteria. Okay, and if they are a spherical shape, means they are known as coccus. The name actually is a coccus, and coccus explains spherical nature. Here you can see this is a singular one. Only single spherical bacteria is then lying singly, not in association with anyone. Then it is known as coccus. But here you can see two coccus are combining, so we call it as a diplococci. What we call diplococci is a plural for the two. Sometimes what happen, they are found in chain. You can see this is the chain. So when they are found in chain, they are known as streptococci, because strip is a term for the chain. So based on that, we can call it as streptococci. Another one terminology is staphylo. Staphylo means like a bunch of a grape. So they are particularly present in a group in such a way that they are known as staphylococci. See, singular is coccus. When two are there, diplococcus. When they are in chain, streptococcus. When they are in a group, staphylococcus. Sometimes they give a 3D structure like this, then they are called as Sarcina, and sometimes they are not present in this 3D form. There's only four are attached, then they are also known as tetrad. So there's different way this spherical shaped cocci bacteria, coccus bacteria can be present. After coccus, another bacteria which are found that are particularly we can say road shape. Okay. Now when we have to talk about this road shape bacteria, so this road shape bacteria are a bacillus. They are technically known as what? Bacillus. So this you can see here road shape bacteria. You can see this diagram. This is the bacillus bacteria. There are several types of bacillus bacteria are there, but they are their shape is actually a rod one. Then another form of bacteria are there that is a particularly one known as Vibrio. We can call this Vibrio as Vibrio as well. Sometimes it is called as Vibrio. So they are comma shape, and for that you can see this diagram. They are looking like somewhat comma shape. Okay, they are looking like somewhat comma shape. Okay. Now next, another form shape you will study that is spiral shape bacteria, and in spiral shape bacteria, if we talk, then they are known as spirillum. Okay, so this four shape of bacteria are found in the particular one. You can see this is a spiral shape, half screw like. They look like what a half screw spiral shape they can appear. Now, if we see this four shape, out of this, remember that some of them have a flagella and some of them do not have. Usually, coccus, if we talk, they are non-motile. If they are non-motile, means they do not have a flagella. So we can call them a non-motile or non-flagellated. Okay, we can call. Okay, we can call it as a non-flagellated. Whereas road-shape bacilli, they can be motile or non-motile. Means some have flagella, some might not have flagella. They can be motile or non-motile. Comma shape also have a flagella, so they are motile. Spirillum also have a flagella, so these are also motile. So this different way they can be formed. So here, usually non-motile are the spherical one. Then road shape may be motile, maybe not, they may have a flagella, they may not have a flagella. But the question which you get from this particular topic is this: which is the most common shape? What question you will get? Most common shape. Majority of bacteria are of which shape? So, and this is the most important question that you should mark. And it is found that most common shape is bacillus, and plural term for bacillus is bacilli. Okay, so this is the different shape which you can observe in case of bacteria. Okay, I hope you understood this in detail. Next to that, we are going to talk about a bacterial structural part, and in bacterial structural part, one thing which we came to know about the cell wall, which is a rigid cell wall, and this rigid cell wall is particularly made up of peptidoglycan. This peptidoglycan is also known as murine and nucleotide, and we have discussed already this. Now, you can see here the bacteria are of two types, gram positive and gram negative. You will study this in cell structure chapter, that what is the actual difference between gram positive and gram negative bacteria. But here we are going to talk about the cell wall difference. Okay. So first thing you should remember about the bacteria, that bacterial cell wall consists of peptidoglycan, murin, or peptide. Two types of bacteria can be there, that is such like gram positive and gram negative. And in case of this gram positive and gram negative, there is a certain differences in this in the cell wall of this. You can see this is the cytoplasmic part, cytoplasmic area. Now you can see in gram negative bacteria, you will find that there is a plasma membrane. In gram positive also there is a plasma membrane which covering the cytoplasm. But above the cytoplasm in gram positive bacteria, you will find a thick layer of peptidoglycan. Whereas in case of particularly gram negative bacteria, two membrane will be found apart from the plasma membrane. That is the outer layer will find that is of lipid, and the inner layer you can see this, actually this is the plasma membrane. Okay, not, it is not an inner membrane of cell, it is a plasma membrane. Okay, and this is the inner membrane, actually inner layer of cell. Now this inner layer is rich in peptidoglycan, but outer membrane is particularly specifically found in the gram-negative bacteria, and it is mainly made up of lipid. So dear students, what you need to remember here that gram-negative have two layered cell wall. As you can see here, this one is the outer layer and this is the inner layer. Whereas the inner layer has peptidoglycan, whereas gram-positive has a single thick layer of peptidoglycan in cell wall. Now, apart from this, we will be dealing one more structure. See, this is the structure part of particularly bacteria. In the structural part, you can see this is a dark blackened particular thing. That is what actually we can say it is a cell envelope, and we will be discussing about cell envelope. But this inner one is the what particularly a plasma membrane, and the infolding of plasma membrane. See, here you can see this is the infolding of plasma membrane, because this is the plasma membrane. So this infolding of plasma membrane is known as mesosome. Okay, this is the cell wall, this is the plasma membrane. Here you can see this is the nuclear material. You can see the flagella, you can see some plasmids. So this is a just picturization image which you by which you can imagine this. Okay, so this is the 70s ribosomes bacteria have. They have a capsule. What are this? We are going to discuss this in detail. Okay, but you have somewhat particularly just imagery diagram of bacteria like this. Okay, so first and foremost thing you should remember that infolding of cell membrane, we remember it is a cell membrane, not cell wall. It is known as mesosomes. Okay, so let's move forward. Next, if we see the genetic material in case of bacteria, it is naked, means it is without nuclear membrane. Okay, genetic material is naked means what? It is not covered by, it is not present in a nucleus, because true nucleus is not there. Here only nucleoid is found, and genetic material is not covered by a nuclear membrane, so it can be called as a naked. Okay, now so here you can see not enveloped by the membrane. Here you can see this is the DNA. Okay, and this is the genetic material. Okay, here you can see this is the DNA. This is which is a genetic material, and it is free. It is not covered by any membrane. So that is what a particular concern with the nuclear genetic material. Apart from this, we will come to know that in case of particularly bacteria, okay, cell organelle are not found in bacteria. As you know that in eukaryotic, see, actually here they have written cell organelle are not found in bacteria. Actually, it should be like a membrane bound cell organ. Which cell organ? Membrane bound. Membrane bound cell organelle are not found in bacteria and in all prokaryotic cell. It is not like that cell organ are not found. That is will be the incorrect statement actually. Membrane bound cell organ are found. And they have recurred cell and same way they have written this statement in another way. The cell organ are not found in bacteria in all prokaryote except ribosome because ribosome are the membrane less. Ribosome are original but they are without membrane. So they are without membrane or membraneless. Because of this, these particularly organelle are found in case of the bacteria. Now let's talk further. Now move to the next term. Motile bacteria contain one or more flagella. Just we have discussed the shape and the motility. If flagella is there, it can help them to move. So motile bacteria have one or more of flagella. You can see here the particularly true flagella, a proper flagella. You can see which is here given. Okay. Now, so this is the what a thing we have discussed. Now, what I want to make you understand that when we draw a particular diagram of bacteria, you will come to know that they have this envelope. But this envelope is not made up of only just one layer. It is a three layer. And when we talk about this three layer, in this three layer, you came to know that the outermost is known as glycocalyx. The outermost layer of this envelope is known as glycocalyx. Then the middle one which is represented here, that is cell wall. And the innermost one is known as plasma membrane. So this glycocalyx cell wall and plasma membrane combinedly called as cell envelope. Now, one more thing I want to make you that understand. The invagination of cell membrane is known as plasma membrane that we have studied. Invagination of plasma membrane that we have just studied that it is known as mesosome. And that mesosome will be found here. So just I want to make rub at this so we can write it proper. These are mesosome. Okay. And apart from this, you will find here a nucleoid content. They have certain plasmids as well. They have plasmids. They have certain inclusion bodies. Okay, styler is not working, might be, but so please forgive for that. But mark this as a inclusion bodies, and this inclusion bodies are important. Now we have to first understand the cell envelope, then we will going to talk about everyone one by one. In this, if we talk about cell envelope, and in this we will talk about what particularly glycocalyx. Okay. So first we are going to talk about, dear students, glycocalyx. As you know that this glycocalyx is the outermost covering which is found in case of the cell envelope, and this glycocalyx is made up of actually non-cellulosic polysaccharide. Okay, it is made up of non-cellulosic polysaccharide. Now it is made up of non-cellulosic polysaccharide, not of a cellulosic polysaccharide, and this glycocalyx can be of a two type. One type it is known as capsule, and another one it is known as slime layer. So what the function of this capsule and slime layer that we have to understand now. In glycocalyx, the particularly there is, if we talk about the cell envelope in glycocalyx, then it is thick and tough layer. If glycocalyx is thick and tough, then it is known as capsule. But if the particularly it is loose and smooth, then it is slime layer. It is smooth, then it is particularly slime layer. Actually, it is came to know that this non-cellulosic polysaccharide include, we remember that include specific polysaccharide as well as some polypeptides are also found that also help in formation of this. Mainly polysaccharide is okay. Now what it helps? Now it is come to know that if certain bacteria have a particular capsule, then this bacteria gives some shiny colonies. Means if suppose there is a petri dish and I am taking a culture medium here, and if I am growing a bacteria on this culture medium, they will give some certain smooth particularly colonies like this. But if I make a particularly, if the bacteria have a slime layer, then you will come to know that when we grow a particular bacteria on petri dish, they will give some rough colonies. Okay, so this gives shiny colonies, but this gives rough colonies. So all rough bacteria have slime layer. Capsule bacteria have shiny colonies. One more important feature we came to know about the capsule that they have particularly virulent property. Those bacteria have capsule, in this capsule have virulent property. So the capsule is the important one for this. Now after capsule, second one is the cell wall. Okay, so next we talk about the, I hope you understood the particular functionality of glycocalyx. Now rubbing this about the particular things about the glycocalyx. Okay, so we can have a particular space to study. So I hope you understood. Okay. So next, inner to the particularly glycocalyx, there is a what, a cell wall. So let's talk about cell wall. Now, when we talk about a cell wall, we came to know that cell wall is made up of peptidoglycan. And this peptidoglycan is a polymer of NAG and NAM. Now, NAG and NAM stands for NAG stands for N-acetyl glucosamine, whereas NAM stands for N-acetyl uramic acid. Now, what happens in case of cell wall? They have particularly chains of NAG and NAM. In peptidoglycan, if we see the structural part, we will come to know that there is a chains of NAG and NAM. And these chains are associated like this. And these are joined by the 4 amino acid. And this 4 amino acid is known as tetrapeptide. This tetrapeptide is making a joining of this type of chains, and the tough structure peptidoglycan will be formed. Okay, so this is the particularly cell wall characteristic. And apart from this, particularly apart from this inner to the cell membrane, cell wall, they have a cell membrane, and we have studied about the cell membrane that it is a phospholipid bilayer structure. And this phospholipid bilayer means it is made up of lipids. So cell membrane of bacteria is phospholipid bilayer structure. And this phospholipid bilayer structure will be look like this. Like this, phospholipids are associated one with another. Some half protein also you can see. Such a way you will find a phospholipid bilayer. Means, suppose this is membrane, we have drawn just this much section of membrane here. And you can see here, these are the, here you can find some protein like this. This protein will be called as extrinsic protein. And this are intrinsic protein. You can see this intrinsic protein are either totally buried like this or they are half buried. But these units are known as phospholipid, and in this phospholipid you will find a head structure. I'm just going for the, you should know that they have a head like structure, this, and these are the tail. These two things are found in so students. So students, here you can see we are talking about head and tail. And this you can see, this is a head region and this is a tail region. This head in phospholipid, specifically if we talk, it is found to be hydrophilic, means water loving, whereas tail is found to be hydrophobic. Okay, so this is the thing about the cell membrane in case of particularly prokaryotic cell envelope. Inner to prokaryotic cell envelope, you will find a presence of nucleoid. Okay, so you will find particularly presence of nucleoid. So if you want to talk about a nucleoid, you have to know that in prokaryote nucleoid is formed. Now nucleoid is the central region which consists of in bacteria double-stranded circular DNA. Double-stranded circular DNA plus non-histone protein, because non-histone protein over which this double-stranded circular DNA overlap, and due to this overlapping, it is known as nucleoid. So sometimes you will find this question that in prokaryotes or in bacteria or in monoras, the nucleoid is made up of double-stranded circular DNA, linear DNA, single-stranded circular DNA, or single-stranded linear DNA. Then you should know that it is double-stranded circular DNA coiled over the non-histone proteins. And the non-histone protein which are used here, they are polyamines. Which proteins? Polyamines. So these polyamines are used here. Apart from this, you will find one more structure from this nucleoid, which is also known as, now this nucleoid is also known as xenophore. It is also known as incipient nucleus. Okay, it is also known as single chromosome. Okay, so these are the things which are used in place of nucleoid. Now, apart from this, we have plasmid. And if you talk about plasmid, plasmid is known as, specifically, if you see, it is extra chromosomal self-replicating double-stranded circular DNA. This is a plasmid. Means, if you see the diagram of a cell, in center you will find a nucleoid, and apart from this you will find some double-stranded circular DNA like this. That is nothing but a plasmid. And this plasmid is extra chromosome, means away from the single chromosome, and they are self-replicating, means they can replicate by their own self. They can form their own copies, and they are made up of double-stranded circular DNA. So this is the thing which is found in case of plasmid. Yes, plasmid are of different types, and in these two types of plasmid are known very well. Number one is F plasmid, and this F plasmid is known as plasmid, and another one that is R plasmid. And R plasmid is resistance plasmid. These two type of plasmid are found in case of the group here, mainly which we study. Okay, we will talk about F plasmid and R plasmid in detail. Now this particularly, if you talk about F plasmid, it is fertile plasmid. Based on this, if suppose this is a bacteria, it has a nucleoid and it has a F plasmid, then this type of bacteria are known as F plus or male or donor bacteria. Whereas another group of bacteria that do not have a F plasmid, suppose they do not have a F plasmid, suppose this have a nucleoid but F plasmid F7, then it is known as F minus or female or recipient bacteria. Okay, and this way this F plasmid help in conjugation. So we will be studying about the conjugation during the reproduction part. And another one is the R plasmid, that is resistance plasmid. And if this is present within the bacteria, it provides resistance to antibiotic. Resistance to antibiotic means the bacteria which have R plasmid can survive in the presence of antibiotic as well. They are not going to be killed. They can survive. So that is the particular type of R plasmid and F plasmid. Another thing which we have studied in case of the particular bacteria, apart from nuclear, that is the inclusion bodies. And inclusion bodies, if we talk in case of bacteria, and in Neet two times they have asked this question, what are the inclusion bodies or what is the function of this? Inclusion bodies are the food storing bodies in case of bacteria. Okay, so these food storing bodies are found in case of bacteria that are inclusion bodies. Okay, and there some further structures are also there. We will be dealing in the further part. So now you have a clear picture somewhat that nucleoid, that if we have a prokaryotic cells have a cell envelope that consists of outermost glycocalyx, then cell wall, then cell membrane, then they have a nucleoid. Invagination of this plasma membrane is lysosome, then they have plasmid are of two types, then they have inclusion bodies, and this all things we have studied now in the structural part. So students, by studying this, we come to know about the structural aspect of bacteria. And in this structural aspect of bacteria, we understood that this bacteria have, if we talk, they have a very simple but very complex behavior. Means the structure is very simple, but behavior is complex. Means the functionality is very complex in case of this bacteria. Apart from this, if we talk about bacteria have a extensive metabolic diversity. Extensive metabolic diversity means we will study in the later part that they have a extensive nutritional diversity. And if they have a nutritional diversity, then they particularly carry out different reaction. This perform several anatomic, the anabolic and catabolic reactions, so they have an extensive metabolic diversity. Apart from this, if we talk about the nutritional aspect, or if we talk about the nutrition aspect of this particular bacteria, then with respect to nutrition, you will come to know that bacteria are of two types, autotrophic and heterotrophic. Autotrophic are those bacteria which can synthesize their own food. But if we talk about heterotrophic bacteria are not able to synthesize their own food. But majority of the bacteria are heterotrophic. Majority bacteria are of which type? Heterotrophic type. In autotrophic also they are chemosynthetic and photosynthetic. We will study what is the difference between these two. And heterotrophic also we talk about parasitic, saprophytic, and one more, that is symbiotic bacteria. Now we will be studying this particular nutritional part in somewhat detail, and we will understand meaning of all these particular terms. So in this term, you will see that particularly the bacteria perform several type of particular nutrition, but some bacteria are autotrophic, synthesize their own food from the simpler inorganic substances, and this autotrophic bacteria are of two types, that is photosynthetic autotroph and photosynthetic autotroph are those which use light as an energy source and synthesize their own food. So here we have to note one thing with respect to the photosynthetic autotroph, and that is they use sun as a source of energy. And sun is a source of energy. Because of this only, this is the term photo is used in the synthesis of food by the autotrophs. Same way, another term is chemosynthetic autotrophs, and chemosynthetic autotrophs use chemical reactions, or they use chemical reaction as a source of energy. So, if they use chemical energy by the any particular oxidation of some inorganic compound happens and the energy get released, if that energy is used in food synthesis, so that's why I use the term chemosynthesis synthetic autotrophs. So we remember autotrophs are those organisms which can synthesize their own food, and they by the using the energy of sun or by the energy or using energy of chemical energy, and based on that the source of energy they are photosynthetic and chemosynthetic autotrophs. And in that also we will be studying about the detail. Now in this, if we see the photosynthetic autotroph, it came to know that this photosynthetic autotrophs use sun as a source of energy, but apart from sun they need a hydrogen and electron donor. If hydrogen is an electron donor, photosynthetic means light is energy source, but here now hydrogen electron, based on hydrogen electron, they are of two types. One is photolithoautotrophs, and photolithoautotrophs in this hydrogen and electron donor are inorganic compound. Whereas another group is photoorganoautotroph, and in photoorganoautotroph H plus and Electron donor are organic compound. Now this H plus and electron react with NAD and then synthesize NADH2. Now this NADH2 is used in dark reaction, and when dark reaction completes, the synthesis of food occurs. So this is the way occur in case of particularly, and here this part is specifically a light reaction, and here it is known as light reaction because light or sunlight is used as a source of energy in photosynthetic autotrophs. So litho means inorganic and organo means organic. Compounds are hydrogen and electron donor respectively in photolithoautotrophs and photoorganoautotrophs. But ultimately photosynthesis occurring like this in case of the photosynthetic autotroph. But another group we have, that is chemosynthetic autotrophs. So in chemosynthetic autotrophs, if we talk, what happens, or in chemosynthesis, if we see oxidation of inorganic compound release, so release energy, and that energy is used in food synthesis. Because of this, they are known as chemosynthetic bacteria. Okay, in chemosynthetic bacteria, so many bacteria comes like bacteria which carry out nitrification. If we need to talk about example, we can write example like this. Suppose NS3 we have, and it converted into NO2 minus, and then it converted into NO3 minus. Now this reaction is known as nitrification. But for this conversion, they needed energy, and during this they release energy, and that energy is used by nitrococcus and nitrosomonas. Whereas nitrate to nitrate converter is carried out by nitrobacter or nitrocystis. So this all bacteria like nitrococcus, nitrosomonas, nitrobacter, nitrocystis, they are all chemosynthetic bacteria. Which type of bacteria they are? Chemosynthetic. And how you will differentiate chemosynthesis from the photosynthesis? In chemosynthesis, energy is obtained by the oxidation of inorganic compounds. So if we go on further and study this in detail, then we come to know that we have the oxidized various inorganic substances such as nitrate, nitrite, ammonia, and use the released energy for the ATP production as we have discussed now. The important statement of NCRT which you should remember here, that is this one, that they play important role in a recycling of nutrients, and which nutrients like nitrogen, phosphorus, iron, and sulfur. And this is the most important question statement of NCRT. So we remember with respect to chemosynthesis. Okay, so this is the important thing. Apart from this, you can see chemosynthetic bacteria include some of the particular one which help in a nitrogen cycle and during the nitrification. Apart from this, if we move further about the nutrition, as, as I told just before some time, that in this majority of bacteria, if we talk, the majority of bacteria are particularly out of the two things, that is autotrophs and heterotrophism. Majority of bacteria are original heterotrophs or heterotrophic. They perform heterotrophic nutrition. So they do not synthesize their own food but depend on the other living organism for the parasite. Now in this, they include first group, that is saprophyte. The saprophyte are the heterotrophic bacteria and that lives on the dead organic matter. That lies on the dead organic matter. Now how they consume food, if that we need to understand, then if there is a one piece of particular inorganic matter, suppose I'm drawing here, suppose this is a piece of inorganic matter and we have some bacteria here. So this bacteria will release enzyme over this and they carry out a breakdown of this. Okay. So this bacteria releases enzyme and then breakdown of this occur. Then this is a particular thing can be carried out by the saprophytes. Means saprophytes are those bacteria which carry out actually decomposition. They carry out actually decomposition. Okay. So that is the thing that performed by the extracellular digestion by releasing the enzymes over the food material. So we have saprophytic bacteria like this. Then majority of heterotrophic bacteria are saprophytes or decomposers. Apart from saprophytism or decomposers, we will be studying about the parasitic ones. So apart from this bacteria can be used in making curd from the milk. Some bacteria are also useful in production of antibiotics. Same way, some bacteria are used in fixation of nitrogen in leguminous plants. So some are symbiotic, some are saprophyte, majority are saprophytes and some are pathogens. When we talk about pathogens, pathogens are nothing but other particular parasites. But these pathogens are causing damage to the human being crops. They can particularly farm animals, they will miss the farm animals. And apart from this, if we can say, they particularly affect the pets as well. Then it is known that so many pathogenic bacteria are there which are positive agents of many diseases such as cholera. And for cholera, you know, Vibrio cholerae is the causative agent for this. For the typhoid, you know, Salmonella typhi is the causative agent. Okay. Apart from this, tetanus is the particularly Clostridium tetani is responsible for the tetanus. Okay, and citrus canker, that is caused by the Xanthomas citri. Okay, so I'm writing the names of that particular all the causative agent of this particular disorder. And in this, cholera is caused by Vibrio cholerae. Okay, then typhoid by the Salmonella typhi. Then if you talk about tetanus, this tetanus is caused by Clostridium tetani, and citrus canker is caused by Xanthomas citri. So these are the causative agent of this particular bacterial disorders. Okay. So next we talk about particularly reproduction. So nutrition, I hope you understood that majority of bacteria are heterotrophic. Now next we are going to talk about a reproduction. And when we talk about the reproduction in case of particular bacteria, we came to know that they perform two types of reproduction, that is asexual and sexual. Now one thing that you should remember here, that in case of reproduction, the asexual reproduction occur, that is present. But in case of bacteria, true sexual reproduction is absent, and they perform another thing, that is sexual recombination or parasexuality. And in asexual reproduction also, either they perform fission in favorable condition. Okay, if all the conditions are favorable, means proper water, proper enzyme, proper sunlight, proper things all are available, then bacteria reproduce by fission. But and in fission, they divide, one bacteria can divide into the two. As you can see here, they perform binary fission, so one bacteria can divide into. But if we talk about unfavorable condition, they produce a one specific type of spore, and they are known as endospore. So they perform specifically asexual reproduction by fission in favorable condition and by spore formation in unfavorable condition. And you can see in this diagram that particularly what is happening. First, it is divided, its nucleus is divided into two, and that will be get converted into the two bacteria. Apart from this, these are the particularly, if you say, this is the structure of spore. And in this structure of spore, if we talk, then or in endospore, you can find that they have an outer membrane, then they have cortex, they have cell wall, inner membrane, and core. Like this particularly spore is present. But if we talk about sexual reproduction, then true sexual reproduction is not present, but here sexual recombination is present. And in sexual recombination, primitive thing occur where a DNA transfer occurs from one bacterium to another bacterium. And this is the diagram which is showing a particular transformation, but we will be talking about the sexual recombination of parasexuality in some more detail. Now, if we talk about parasexuality, dear students, if we talk about parasexuality or sexual recombination or sexual recombination, here primitive type of exchange of genetic material occur. And this sexual recombination is of three types, that is transformation, then conjugation, and transduction. These are the three methods of particularly sexual recombination or parasexuality. Dear students, this transformation experiment which is performed by Frederick Griffith scientist on E. coli, that we are going to study in class 12th. Okay, and how Griffith performed this experiment, that we will study there. Transduction also, it is a process in this transfer of DNA from one bacterium to another bacterium with the help of viruses, and it is known on transduction. That is transfer of DNA segment from one bacterium to another through bacteriophage or virus, then it is known as transduction. Suppose there is one bacteria A, it has its own nucleic material, it has its own plasmid, and another bacteria we have that is B bacteria. And if some DNA is taken from here with the help of bacteriophage and bacteriophage transfers genetic segment of DNA from here to here, then this process is known as transduction. Okay, but for us, conjugation is somewhat in detail we have to study, so we will be dealing at last. But we remember transduction was performed by Zender and Liderberg on Salmonella typhi. Whereas next we are going to talk about conjugation, and we will discuss about conjugation in detail. And conjugation is given by Liderberg and Totem, and this conjugation studied on E. coli. Okay, so we will be dealing with that in detail. If we want to talk about conjugation, then first we have to talk about the F plus and F minus bacteria. Now in a F plus and F minus bacteria, if we talk how they perform function, that we have to understand. So what happens? Suppose this is a F plus bacteria. Now if F plus bacteria is there, then it should have F plasmid. And if there is a F plasmid, then they have one gene that is known as F factor or F gene. Now this F factor or F gene, if you talk, has an ability or a information for the pillin protein. And if it has a information for the pillin protein, it can form pilus or pili, which come from cell wall like this, which come out like this. So this is the pilus. Now it conjugate with another one which do not have a plasmid. They have nucleate but a plasmid is absent. That is F minus or female or recipient bacteria. Now what happens? This combined with this they have F plus and F minus bacteria. Now what happens? This is a nucleoid. This is a nucleoid, and you know that this is the F plasmid. But F plasmid definition we have studied that plasmid is autonomously replicating. So what will happen? The plasmid will be copied automatically. There are several F plasmids there, and one of the F plasmid passes to the F minus. So now, what will happen after the breakdown of this conjugation bridge? This is what? What is this? This is known as conjugation tube or bridge. And when the breakdown of this occurs, These two bacteria are separated. These have nucleoid. This already have plasmid, so it is F plus. Plus another bacteria we will have that have a nucleoid as well as a plasmid, and that also become F plus. So for this, if we write the equation of this particular will be like this, that when F plus conjugate with F minus, they give rise to F plus plus F plus bacteria. And this you can see proved by this one. But what happened? This is a plus maintenance plus bacteria, which was earlier it was F minus. So here one thing happened that F minus converted into F plus, and the conversion of minus bacteria into F plus bacteria due to transfer of this one. This is known as sex reduction. So this process is conjugation, where a transfer of genetic material occur or a plasmid occur from this particular one bacterium to another bacterium through the conjugation tube, then which is known as conjugation. And it is found in case of bacteria during the sexual recombination. But the most common is asexual, and in asexual also most common is which one? Binary fission. So dear students, I hope you understood this particular reproduction part in case of bacteria. Apart from reproduction, next we will be dealing further some of the things with respect to particularly bacteria. In bacterial structural aspect, we have missed one thing that we have to discuss here. The structure flagella. Pilus used to understood. Now we have to need to understand the structure of flagella. When you see the structure of flagella, you will find that there is a presence of two rings in the cell wall. And in that one rod was inserted. Your hook will be like this, and like this filament will be there. So this is a filament of flagella. This is hook. Here you will find ring like L, P, S, and ring. This is a cell wall. This is cell membrane, and in this is rings are inserted. So this whole combinedly known as basal body, and these three combinedly form a flagellar structure. Now in eukaryote, in cell structure, you will study that there is several microtubule and tubulin protein form a filament. But here this is the prokaryote bacteria. In prokaryote filament is made up of flagellin protein. Which protein? Flagellin protein. So you should remember this concept. It is important one. And when you will study cell structure, you will remember this particular part. Okay, so this is the structure of flagella. And only one single strand of tubulin can be present. So sometimes this filament is known as single stranded or monostranded. Okay, so this is the particular thing of flagella. We have already understood the pili, and pili help in conjugation. Flagella, if we talk about function, then flagella help in locomotion. Okay, I hope you understood this in detail. Now what we will do, we will be dealing with the particular part, that is fimbriae. Sometimes, yes, we will talk about this, but we should understand about the fimbriae. So sometimes over the bacterial body, we'll find some short bristle-like appearances over the whole body, and these bristle-like appearances over the body of bacteria are known as fimbriae, and these fimbriae have been attachment. Okay, so this is the thing. Okay, so now the structural part is over, nutrition part is over, reproduction part is over. Okay, that all thing we have discussed in detail. So next we will be talking about classification. So dear students, till now we have discussed the structural part of bacteria or monorems. Then we have dealt with the already particular some functional life processes. And now we are going to talk about a classification or different types of bacteria which are found in kingdom Monera. And in that, first we are going to start with cyanobacteria. Which bacteria? Cyanobacteria we are going to start. Okay. So dear students, if we see cyanobacteria, they are also known as blue-green algae. They are known also as blue-green algae. And if we talk about this blue-green algae, they are also known as blue-green algae. You can see this is a pictorial diagram of a cyanobacteria and they are appearing in a filamentous way. Now, if we talk about cyanobacteria, first before understanding this some of the important points, if we go in the history of classification, according to two kingdom, Linnaeus placed this blue-green algae in Cyanophyceae. So, so let's talk about that particular part. Now, if we talk about cyanobacteria, according to two kingdom classification, Linnaeus placed this particular cyanobacteria in the Cyanophyceae or Mixophyceae class of algae. But if we talk about now, due to the particularly prokaryotic structure, they are placed in Monera. Now, dear students, this cyanobacteria have a chlorophyll similar to the green plants. So, if we talk about a particularly pigments, they have chlorophyll A. Okay, and due to the presence of chlorophyll A, they can perform photosynthetic. So, these are the bacteria which have an ability to synthesize their own food. They are photosynthetic autotrophs. Apart from that, when we talk another thing, they are unicellular, colonial, filamentous, aquatic, or terrestrial. Okay. So, dear students, I want to make you understand that if we are going to, we are talking about the pigments. This cyanobacteria have chlorophyll A. Okay. Apart from they have a carotenoids. They also have carotenoids. Okay. If we talk about chlorophyll A, you know that chlorophyll A is green in color. Whereas if we talk about carotenoid, that carotenoid are somewhat yellow. And apart from carotenoid, some phycobians are also found. Some phycobilins are also found, and that phycobilins can be blue or red in color. So usually this particularly a blue-green color is due to the pigments and due to presence of chlorophyll A, which is similar to plant, they can synthesize their own food. Apart from this, you can see here they can be a unicellular, means they are made up of single cell. They can be colonial or they can be filamentous. So they can be unicellular, they can be in a colony, okay, or they can be a particularly filamentous like this in a form of filamentous. They are found in aquatic or terrestrial algae. They are aquatic or terrestrial algae. But if we talk about the habitat, if we talk about the habitat in aquatic, they are freshwater mostly. They are freshwater mostly. So mostly, usually, they are found in freshwater. Okay, some are found in marine water as well as they have a terrestrial habitat. Okay, so these are the some of the diagrams of particularly cyanobacteria. You can see here there is a chlorococcus. Okay, then you can see the Pleocapsa, then Oscillatoria, then Tolipothrix, then Merismopedia, and Geotrochia. Now in this, you need to remember only this one that is important with respect to our syllabus, and you can maximum remember this one. These two are important with respect to our syllabus and according to NCRT. Okay, dear students, so this is a primitive information about cyanobacteria. Now, these are the most primitive organism and they show particular oxygenic photosynthesis. Okay, so next we are going to talk that initially, the bacteria was performing an oxygenic photosynthesis, means the oxygen was not released, but they evolved into the blue-green algae, and blue-green algae was the first organism to carry out photosynthesis and release oxygen. So they have a capacity of release of oxygen, so they are oxygenic. And due to the capacity to release oxygen, several aerobic forms arise due to this organism. Okay, so you know that in light reaction, light comes due to the use of water, oxygen evolves, and after light reaction, products of light reaction are used in dark reaction, that is Kelvin cycle. It is a part of dark reaction. Here CO2 is used and sugar is synthesized. So here you can see we will study in detail in chapter photosynthesis, how photosynthesis occur in detail. But here this is giving a just preliminary glance. And here you can see it is explaining a light reaction and it is explaining a dark reaction. In light reaction, what is happening? Light is coming, that is accepted by pigments, and in chloroplast or thylakoids, water is used and O2 is released. The products which are obtained by the light reaction like NADH2, FADH, NADH2, NADPH2, and ATP, these all products are used in dark reaction, Kelvin reaction, and in Kelvin cycles, CO2 fixation will occur, and after CO2 fixation, sugar formation will occur. So this is explaining us simply how photosynthesis occurs. Now, if we see the internal anatomy of a particular one cell of this either unicellular, filamentous or colonial blue-green algae, you will find that they have a particular structural component in the protoplasm. And you will see this type of diagram in the protoplasm. In this protoplasm, you will see that the centrally they have located circular nucleate because yes, bacteria have circular DNA. Some protein granules are also there. They have a ribosome, which are 70S. They have some photosynthetic lamellae similar to thylakoids, which have performed photosynthesis, and they have a particular pigments over this. Plasma membrane is there, cell wall is there, and all over the cell is covered by this covering, and that is gelatinous coat, and we call this gelatinous coat as mucilaginous covering. we call as mucilaginous covering or gelatinous sheath. And this mucilaginous covering or gelatinous sheath is found around all over the body of an organism. Now, if you see, the lamella are arranged periphery and pigments are arranged in the periphery, but centrally there is only DNA. Pigments are not located in the center. So what happens due to this? The protoplasm of center, that is central protoplasm, that become colorless, and that central colorless protoplasm is known as centroplasm. But and that is with chromatin material. Chromatin material is not going to give any color, but peripheral pigmented chromoplasm is there. Why it is chromoplasm? Because they have pigment and that pigment give color. So this chromoplasm is colored. So due to this, the cytoplasm is divided into two parts in case of this blue-green algae. Okay, so I hope you understand and getting these points one by one. We will be dealing one by one points in a detail, and slowly we are going to understand all this. So I hope you understood till this. Now let's move further. If we talk further, you will find that the filaments or colonies, if you find, that's surrounded by the gelatinous sheath, if you see. Now here one more term you are getting, that is trichomes. These trichomes are nothing but filaments. If this is a particular filaments or particularly blue-green algae, then they are known as trichomes. One filament is known as trichomes in this. Now you can see the filamentous nature here, gelatinous sheath which is covering here. You can see in the pictorial diagram the whole the filamentous bacteria blue-green algae is there, and this filamentous blue-green algae is covered by a gelatinous sheath. Yes or no? Which we can call also what? Mucilaginous sheath. Okay. So as we move on, sometimes it is found that this particular blue-green algae grow very rapidly in a polluted water. And when they grow in a very rapidly in a polluted water, they form water blooms. So this is the important statement. It was asked earlier in Neet. It is a water bloom and it is formed in the polluted water. Okay. Now, then you can see this is the whole particular diagram of blue-green algae or algae showing, and that is algal bloom. Actually, it is a diagram of what? Algal bloom. Yeah, we can call it as a water bloom. What we can call water bloom. Yeah, or algal bloom. Now, next to that, you can see that the red color of Red Sea. Now, one concept you have to remember that sometimes you will find a reddish coloration in water of sea, and that reddish coloration is due to one blue-green algae. And the name of that blue-green algae is Trichodesmium erythraeum. This Trichodesmium erythraeum gives a red color to the sea, and due to we call this concept as a red sea. We will study red tide in protista, but here it is given red sea. So the direct question you can get from this: who is responsible for the red sea? So this is the bacteria Trichodesmium erythraeum blue-green algae. You can see here this is the reddish coloration due to the overgrowth of this Trichodesmium erythraeum. Apart from this, we come to know that in some of the particularly cyanobacteria, they, apart from carrying a photosynthesis, they also have an ability to fix a nitrogen. But for fixing the nitrogen, they need a special cells, and this specialized cells are known as heterocyst. They are known as what? Heterocyst. So we have to talk about this heterocyst in detail, and we will discuss that now. The cyanobacteria in which you will find a nostoc, the heterocyst, that is nostoc and anapina. So here we can write that this nostoc and anapina are heterocystous bacteria. They are heterocystous bacteria. Okay. Now, the particularly other bacteria which lack this heterocyst, they are homocystous. Okay. Now, you can see here in a diagram that this is a thick-walled specialized cell which is known as heterocyst. This is heterocyst. Okay. And we will study the structural detail with the heterocyst. Here also one detailed, one enlarged view you can see this is a particularly heterocyst. You can see this is an enlarged view of heterocyst. Okay. Now what are the characteristic feature of heterocyst that we are going to study? And in that characteristic feature, first we will understand that how heterocyst work. Okay. Suppose this is a filament. It is covered by a gelatinous sheath all over the body like this. But one cell is there which is a thick wall and the gelatinous sheath is absent over that thick cell. This specialized thick cell is known as heterocyst. Dear students, so if we write the characteristic of heterocyst, we come to know that heterocyst is thick walled. They have a thick wall. And this thick wall are impermeable to oxygen. So oxygen cannot enter this. Apart from this, they have PS1 only. PS1 is nothing but photosystem, and photosystem is nothing but a group of pigment. It is known as what? Group of pigments. Now this group of pigments are of two type. Like photosystem are of two type. PS1 and PS2. Actually with PS2 oxygen evolution occur. And if PS with PS2 oxygen evolution occur, so this PS2 is absent. So if PS2 absent but PS1 is present, so oxygen evolution does not occur. And when oxygen evolution does not occur in the heterocyst, nitrogenase enzyme become functional. Get functional. Get functional. When nitrogenase enzyme get functional, this nitrogen enzyme carry out conversion of nitrogen into NH3. That is ammonia, and this is known as nitrogen fixation. So this heterocyst have a capacity of particularly we can say a nitrogen fixation, thick wall, and due to the thick wall nature and the absence of PS2, anaerobic condition are maintained within the heterocyst, and this heterocyst carry out nitrogen fixation. Important thing that you should remember that heterocyst lack gelatinous covering. Okay. So I hope you all are understanding this in detail. and this are the normal vegetative cell. Now, how this vegetative cell is differentiated from this specialized heterocell cell? This vegetative cell has both PS1 and PS2. So, if PS2 is there, they can carry out of oxygen evolution. And so, this oxygen is responsible for evolution of aerobic forms. Yes or no? So this is the thing with respect to the heterocyst. Okay, dear students, I hope you understood that what is heterocyst, what is the specialized cell, and I hope you all are clear. Now we have to study some of the points with respect to this. Okay, before that we have to rub this. Okay, I hope you have written this, so I am removing this particular part, and then we will study its particular point. Okay, so now let's talk further. So now, after studying the particular internal single cell structure, their way of arrangement, their trichomes, their unicellularity, their pigments, their photosynthetic nature, then the nitrogen fixing nature, and for nitrogen photosynthesis, we have studied that they are the first photosynthetic, oxygenic photosynthetic carrier on the trio. So on Earth, these are the first organism we produced oxygen on Earth. Now, we will be talking some detail with respect to reproduction in case of this particular cyanobacteria. And when we talk about reproduction, we come to know that they perform asexual reproduction. And in asexual reproduction, cyanobacteria form a specialized structure known as a hormogonia. They are going to form a hormogonia, or they are going to form a thick-walled echinoids. Why? Because typical sexual reproduction is absent in case of this cyanobacteria, and this is the one of the important statement which can be possible to ask in exam. Okay, so now let's talk about hormogonia and thick-walled echinoid. One more thing you should remember, that flagella is absent. This statement they have asked earlier in Neet. Important statement that flagella is absent throughout the lifespan. Either it is a vegetative stage or a reproductive phase. In any part, any phase of life cycle, you will not find a flagella in this. So means the cyanobacteria are non-motile. Okay. So very good about the important point with respect to that. So this is a particularly hormogonia. Okay. So, if you want to understand what is this hormogonia, okay, then we can understand that these are the particularly hormocysts, and by formation of this one thick-walled cell, what you will find that this a heterocyst there, this is a dead cell. This is a thick-walled dead cell. If dead cell get formed, the filaments get separated, okay, and the filaments will particularly survive. But another one is the hormocyst. Hormocyst also break this filament into the two particularly different conditions. This echinoid specifically formed in unfavorable condition. That you should have to remember. This echinoid formation occur in unfavorable condition. Okay, and this, if we talk about hormocyst, these hormocysts are found in oscillatoria. Formation occur in case of oscillatoria. And if hermocyst formation occur here, this two filament will be acting as a new organism. Same way, if akinete form in unfavorable condition, okay, then these are the dead cells, and these dead cells will degenerate in the part. Okay, but what degeneration also can occur, but what is happening? They store food and they remain inactive for the unfavorable condition. Whenever the favorable condition will come, they will germinate and they will give rise to the new individual. Now, apart from this, after cyanobacteria, yes, before understanding the mycoplasma, we have to understand one more thing. They also perform binary fission and fragmentation. They also perform binary fission and fragmentation. If you talk about binary fission occur in case of unicellular forms only. Means single cell forms can perform binary fission, but fragmentation you will find in case of filamentous form. Okay, so this is the thing that you should remember with respect to the reproduction. But they mainly reproduce by asexual reproduction. There will not be any particularly asexual reproduction. Apart from that, next is mycoplasma. Okay, but I want to make you understand something about this particularly. You can say some of the importance of gluconeogate that we will study in detail. Next, we talk about the mycoplasma. This mycoplasma are the bacteria in which cell wall is completely absent. So, the direct question from NCERT you can get the wall-less bacteria are nothing but which one? Mycoplasma. In this wall-less bacteria, you will come to know that due to absence of cell wall, due to absence of cell wall, what is happening? Plasma membrane becomes the outermost covering, and a plasma membrane is outermost covering. So what will happen? They can attain a different shape. So this mycoplasma have variety of shape, and due to the capability of presence in variety of shape, they are known as a pleomorphic. Then, so here you can see shape of the organism not constant or exist in a various shape. Then, the one more important thing about the mycoplasma is they can alter their shape as well. They can change their shape and size in response to the environmental conditions. Now, one more thing that you have to remember that these are the smallest living cell. Okay, they are the smallest living cell because they are also known as PPLO. And if we talk about the size of this particularly mycoplasma, it is less than zero point 0.3 micrometer. It is less than 0.3 micrometer. So they are smallest living cell, but they can survive without oxygen as well. So the bacteria which can means it is showing a possibility they can survive without oxygen means usually they are particularly aerobic means they need oxygen, but sometimes they can survive without oxygen. So they are actually a facultative anaerobe. They are facultative anaerobe means sometimes they can survive without oxygen, but usually they need oxygen. So that is a mode of respiration in case of the mycoplasma. Apart from that, if we talk about this mycoplasma, it was initially called as PPLO. Now, why it is known as initially PPLO, that is pleuropneumonia-like organism, because they were discovered from the pleural fluid of cattle suffering from the pleuropneumonia disease. Discovered from where? pleural fluid. You know that pleural fluid is present in between the outer and inner membrane of the lungs. And if this pleural fluid is present and it is infected with the disease of the kettle of the from the kettles of the infected with the infected with the disease, that is pleuropneumonia. Because of this, they are named as pleuropneumonia-like organism. Okay. Now, this is the structure of this particularly mycoplasma. And when we talk about the structure of mycoplasma, you can see here the particularly cell wall is not here, and the lipoproteineous plasma membrane is outermost covering. Okay. Apart from this, they have a DNA. You can see they have a linear DNA, double helical, double stranded linear DNA. Okay, they have a double stranded linear DNA. Apart from this, they have ribosome. That ribosome are 70S. They have RNA. They have metabolites. Now we remember this plasma membrane is trilamellar, means three layered. This plasma membrane is three layered. Three layered, particularly plasma membrane is found in case of this. Means that in this cell wall is absent, so we can call this also as a bacteria with their coats of, we can call bacteria with their coats of, we call them BPLO. We also call them jokers of plant kingdom. What we call jokers of plant kingdom. Why we call jokers of plant kingdom? Because earlier it was placed in plantae according to two kingdom, and due to the absence of cell wall they can gain a different shape. And you know that what joker do, they have a different, particularly different shape or different images, or due to this, these are also known as jokers of plant kingdom. Okay. Next, we found that, or it was known that they affect both plant and animal. Okay, they affect both plant and animal, so they are pathogenic to plant and animal. Okay, one more question I want to make you understand from the previous slide, that we in diagram in the previous slide we have seen that they have DNA and RNA both. So if the question is like that, which organism have DNA and RNA both, then you will write a mycoplasma. But if the question is like this, that which have a DNA or RNA, then it should be viruses, because viruses have either DNA or RNA. But mycoplasma has both DNA and RNA. Okay, so they are can be pathogenic to plant and animal. Example, if we see witch's broom, that's the disease which is caused by mycoplasma in case of plant. Okay, and you can see this is a particularly a witch's broom disease in case of the plant. Now, sometimes in animal they cause particular disease that is known as pleuropneumonia. The name was given based on that initially. So you can see this is a particular cattle affected with the pleuropneumonia. Okay, weak, and they was not able to perform their body functions properly. Then mycoplasma also cause urethritis in human. The urethra get damaged. Okay, so this is the thing. Now I want to make you to write some of the important points with respect to this mycoplasma, and these are like this, that this mycoplasma was discovered by Knockard and Rocks. Now, if we talk about Knockard and Rocks, these are the two French scientists. In 1898, they have discovered this particularly while studying the pleural fluid of cattle suffering from pleural pneumonia, as we have discussed. But later on, if we say Novak was the person in 1929 put this organism under genus Mycoplasma. Okay, then if we talk later on, they are also found to be known as that amylose, that is molecules like organism. Molecules like organism or mycoplasma like organism, they are also known. Now, one Japanese scientist, Doi et al., discovered that this mycoplasma also cause one ester yellow disease. Which disease? Ester yellow in case of plant. And when they cause this ester yellow disease in case of plant, phloem cells of plant are much affected by disease. So we remember in ester yellow, phloem cells like sieve tube and companion cell, sieve tube and phloem parenchyma. Sorry. Why it is not working? Yes, now it is working. Again, it is making a problem. So phloem parenchyma was the particularly, we can say here, response are affected by these cells. Means ester yellow phloem get affected by this one. These are unicellular, they have DNA, RNA, all this we have discussed. Now, one more important feature that we have to discuss and that is important here, that this mycoplasma are insensitive to penicillin antibiotic. Now, why they are insensitive to penicillin antibiotic? Because they lack cell wall. And penicillin inhibit cross linking of peptidoglycan chains. And due to inhibition of cross-linking of peptidoglycan chains, they are insensitive to penicillin because they do not have a cell wall, so penicillin cannot act on the cell wall. But they are sensitive to tetracycline and chloramphenicol. Why? Because this tetracycline and chloramphenicol antibiotic acts on a metabolic activities. Okay, then they are known to cause plant and animal diseases. And the reproduction, if we talk, the detail about the reproduction is not studied much more. But in a reproduction, they either perform binary fission and which is the most common method. But some scientists have shown that elementary bodies like structure somewhat found in case of the cell of this mycoplasma, which are helping in a reproduction. They have a role in reproduction. It was discovered, but how they are helping, that is not known till yet. And in plants, one more important point you have to note down, that Mycoplasma for plasma disease are transmitted by leafhopper. Mycoplasma disease are transmitted by leafhopper. That you should remember. Okay. So these are some of the important questions and important things all about the mycoplasma that we have discussed. Okay. Now I hope you are clear with this mycoplasma. Next, we are going to talk about another group of bacteria, another group of eubacteria specifically, that is actinomycetes. Dear students, these actinomycetes are, when grown on the petri dish, you will see such type of diagrams. Is you will see some of the radial colonies like this. You will see radial colonies like this. Due to the radial colonies, this actinomycetes are also known as ray fungi. They are also known as ray fungi. Okay. Now, so they have a branched filamentous. They are branched filamentous bacteria. They are also known as filamentous bacteria, and they form radiating colonies. So they are also known as ray fungi, as we have discussed in the culture. Now, one more important feature that you have to remember, that the cell wall of this actinomycetes has peptidoglycan but also has a mycoplasma. So it is not that they have mycoplasma only, they have peptidoglycan as well, or murine as well. So you should remember this point as well. Okay. Now, apart from this, if we talk about this particular actinomycetes, most of them are saprophytes or decomposers. They can carry out a decomposition mainly. But some of them act as parasitic as well, and we are going to talk about one example of some of the actinomycetes. They are Mycobacterium and Corynebacterium. Okay, you can see the diagram of Mycobacterium. This Corynebacterium. These all are filamentous bacteria. Now, one more thing was found in case of this actinomycetes is that these are the first born on our planet, and they are present nowadays with a primitive character. It is considered that these actinomycetes are, particularly if we see specifically, then they are a connecting link between bacteria and fungi, because they have a mycelial body and they have a prokaryotic structure. So they are also known as mycelial bacteria. They are also known as mycelial bacteria. Now in actinomycetes, so many antibiotics are obtained from a genus, that is Streptomyces of actinomycetes. Apart from this, several examples are included in this particular one. Some more example you should note down, like Frankia. We have just studied the example Mycobacterium, Corynebacterium, Streptomyces, but Frankia is also one of the known example, as Frankia is a filamentous bacteria or actinomycetes. This show symbiotic association with roots of non-leguminous plant for nitrogen fixation. Dear students, we remember that Non-leguminous means what? They are not belonging to the leguminous family. They are not belonging to the pulses. They are not a gram. They are not like this. Okay, they are not a soybean, belong to the soybean family. Then they are a non-leguminous plant, and it shows symbiotic association, mean mutually beneficial association with this roots of this for nitrogen fixation, and they carry out this nitrogen fixation by nodule formation. By nodule formation. Okay, so that is the one of the clear picture of this particular actinomycetes. Okay, so here they are also particularly telling that first they rub this point. The number of antibiotic are produced by the actinomycetes and the member especially streptomyces. Just we have discussed this is the streptomyces diagram. Okay, so this is all about actinomycetes, and now we are going after completion of this eubacteria. Okay, students, till now we have completed eubacteria, and eubacteria we have studied cyanobacteria, mycoplasma, and actinomycetes in detail. Now we will be talking about the archaebacteria. Eubacteria are normally found everywhere, but archaea are found in some of the extreme condition. Archaebacteria are found in some of the extreme condition. Now, one statement I was used wrongly that actinomycetes are primitive bacteria. Actually, they are not. Archaebacteria are the primitive bacteria, and these archaebacteria are considered as oldest living fossil. Which one? Oldest living fossil. Why they are considered as oldest living fossil? Because majority members are not found, only few are found, and still they are living. So because of this, they are oldest living thing. Evolutionary, they are most primitive, and they have a characteristic, or they are special because they live in some of the most harsh habitats. And you can see this is the diagram of some of the archaebacteria known in the microscopic view. Now, they are of three types. Some found in extremely salt area, they are halophiles. They are known as halophiles. They are of three types: halophiles. Then they are belonging to the another group. Some of them which are found in hot spring, they are thermoacidophiles, and some which are found in a different condition, marsh area, they are onisimals. Okay, so we will be talking about them in detail. Okay, but before that, we understand how archaebacteria differ from the other bacteria. Okay. So what are archaebacteria? Found in harsh habitat. Which archaebacteria are found? We have discussed just three types. Now first we compare the archaebacteria with the bacteria because they have a different cell wall structure. And in different cell wall structure, this archaebacteria do not contain peptidoglycan, but they have the pseudomurine. And this pseudomurine is chemically known as NAT, that is N-acetyl allose uronic acid. Okay. Actually, there is no need to remember the chemical nature, but you should know this is for the information that N-acetyl allose uronic acid is nothing but pseudomurine. So you can see here, in case of particularly archaebacteria, cytoplasmic membrane is there. Okay, in cell wall, upper the cytoplasmic membrane in cell wall, they will find a pseudomurine. Above that, you will find a S layer. Okay. Here also you will find another two diagrams are given: cytoplasmic membrane and S layer. Okay, that is a peptidoglycan layer. But here you will find a pseudomurine as well in case of the particularly archaebacteria. Now, one more important difference with respect to cell membrane among archaebacteria compared to eubacteria is cell membrane have a branched chain lipids. And these branched chain lipids are found in cell membrane, not in cell wall, we remember. And they have phytanyl side chain. And this phytanyl side chain is nothing but a methyl side chain. And one more important is that branched chain lipids decreases membrane fluidity. And due to decrease in membrane fluidity, they give a somewhat rigidity to this archaebacteria, so they can survive in some of the unfavorable conditions. Okay, so survival in extreme condition, that is one of the important feature. Something we are going to talk about the types, three types of bacteria. In that, we are going to talk about methanogens and which are the first bacteria. If we talk about methanogens, where they are found in the gut of several ruminant animals like cows and buffalo. If you see, they have another. I mean, zoology might be studied that they have another part in the stomach that is known as rumen. And in rumen, these bacteria are found because in cattle or in cow or buffaloes, there is a need of cellulose digestion, which come in the plant material, and for this, these bacteria are necessary. So why they are known as methanogens? Because by breakdown of this cellulose, they release methane. So the producers of the methane gas, so they are known as methanogens. Okay. In this, we will discuss some more things about the particular methanogens, halophiles, and all this in the particular one. Okay. So let's talk. In methanogens, it came to know that they are methane producing. They are found in marshy area. It is all are correct. They produce methane. That all are correct. But the example of some of the methanogens are necessary to understand, and that are methanobacterium. Another example is methanococcus. Another example are methanomicrobium. But the bacteria which is found present in the gut of ruminant organism or in their rumen is known as rumenococcus. Now, if we talk about more detail in methanogens, if we talk about respiration and nutrition, we came to know that the respiration is obligate anaerobe. Means they cannot survive in the presence of oxygen. And apart from that, if we talk about nutrition, in nutrition you came to know that they are chemoautotroph. Okay, they perform chemosynthesis. So that is the first bacteria, that is methanogens. Apart from methanogens, the another group of bacteria that are found in case of this archaebacteria, that are halophiles. So let's talk about something about the halophiles in detail. Now, when we talk about halophiles, these halophiles are found in salty areas. And if we talk about these halophiles, one important feature that you need to remember, that is their example, and that is a halococcus. They are found in salt containing area, definitely. But if we talk about their respiration, it is known that they are facultative anaerobe. They are facultative anaerobe. And if we talk about particularly nutrition, they are heterotrophic. Now the question is why they are heterotrophic? Because it is known that in case of halophiles, when there is a too much light availability or high intensity of light, they synthesize over a membrane that is known as purple membrane. And this purple membrane receives energy and form ATP. But this ATP is not used in CO2 fixation. So they cannot synthesize food. It is used in several other activities. Okay, it is used in other metabolic activities of the body. So they cannot synthesize their food, and because of this, they are heterotrophic. Okay, so you should remember this respiration in nutrition. And after discussing these methanogens and halophiles, we are going to talk about the last one, but not the least, that is last one, that is a thermoacidophiles. Okay, so first we should remove this and then we will discuss. Now, let's talk about thermoacidophiles. Dear students, as the name suggests, thermoacidophiles, they can be found in a high temperature area and low pH. They are found in high temperature areas, and high temperature is approximately, we can say, 80 to 100 degrees Celsius. And if we talk about pH, then pH is low, and the low pH is up to the pH 2 they can survive. So they are the bacteria which are found in hot water springs. Now we can talk about this for the particularly thermoacidophiles, that is the respiration and nutrition. Then we come to know that respiration, they are obligate anaerobe, and if we talk about nutrition, they are here chemoautotroph. So out of these three bacteria, obligate, sorry, they are, these thermoacidophiles are not obligate, they are facultative anaerobe. Okay. Okay, we have tried this, what? Facultative. Is my mistake. So till now, whatever the three types of archaebacteria we have studied, methanogens are the only obligated anaerobe, other two are the facultative anaerobe. Okay. And with respect to nutrition, thermoacidophiles and methanogens are chemoautotrophs, whereas halophiles are heterotrophs. Okay. I hope, dear students, you have understood this archaebacteria in detail. So let's move further and talk about some of the questions. Dear students, we will be dealing with some of the questions in detail about this bacteria, this monogens, and you will have a thorough understanding of this topic by the dealing with these questions. So let's start dealing of questions. First question we are going to deal that bacteria which live under extreme hot conditions. Just we have discussed about the archaebacteria. So they can be acidophiles, they can be thermoacidophiles, methanogens, or none of this. But you know that here hot condition, extreme hot condition. So obviously they are the bacteria which found in high temperature, and they are thermoacidophiles. Next question we are dealing that the infolding of cell membrane are called. We have discussed this. Okay, that if plasma membrane is there or cell membrane is there, this cell membrane will form one infolding, and this infolding is known as secondary structure. No. Mesosome, yes. Chromatosome, none of this. So they are mesosome. Now one more question you can find along with this mesosome, that what are the functions of this mesosome. So we remember mesosome help in respiration. That is the first function, and second function is replication of DNA. Why? Because they are associated with the nucleoid and they help for the replication of DNA during the binary fission. So mesosome is the correct answer, is the infolding of cell membrane. Next to that, We are going to understand one more thing. We have discussed this heterocyst in case of blue-green algae. So heterocyst relates to the following function. First is carbon fixation, another one is a nitrogen fixation, then phosphorus fixation, then magnesium fixation. And you know that we have studied it in detail that how they carry out a nitrogen fixation. And we have also dealt with the characteristic. Next is spore or endospore are formed during, you know that. Bacteria when have a favorable condition they undergo asexual reproduction through binary fission, but when they have an unfavorable condition they undergo endospore formation. So the answer should be unfavorable condition. Okay, easy questions. Then one more thing, important one. A peculiar characteristic of actinomycetes, the cell wall of actinomycetes contain uric acid, mycolic acid, lactic acid, glycolic acid. And you know that we have understood it is made up of mycolic acid and peptidoglycan. So mycolic acid becomes the correct answer. Then we move to the next question, that which is not a actinomycetes member among the following, which is not a actinomycetes member among the following. So if we see the example, mycobacteria are obviously an example of actinomycetes, that is filamented bacteria or ray fungi. Corynebacterium is also there. Corynebacterium leprae, I hope you understood. Streptomyces we have dealt, that is also found in actinomycetes. And last in Trichodesmium. Dear students, we have studied that Trichodesmium erythraeum is a blue-green algae, and this blue-green algae is responsible for the Red Sea. So it is not a particularly actinomycetes, so it becomes the correct answer. Okay, dear students. So let's move to the next question. The type of organisms that play a great role in recycling of nutrients. Dear students, this is a direct NCERT based question that you have to remember, and they have asked several times this particular question. So you should remember these are nothing but chemosynthetic bacteria. So first is photosynthesis, second is chemosynthesis, third is pathogenic, and fourth is the symbiotic. So answer should be chemosynthesis. Next to that, we move to the another question, that monomers include, and you know it is easy use question. They include bacteria, blue-green algae, fungi, lichen, and all prokaryotes. Actually, all prokaryotes are included in the monomers. So most appropriate answer becomes fourth. Next question is monomers specially live in most harsh habitat are, that are not eubacteria, they are archaebacteria. Easiest question. So second will be the correct answer. Which of the following is not found in monomers? Yes, we have to select which of the following is not found in monomers. So first option is DNA. We have studied the structure and we know that in bacteria they have a double circular DNA. So it is there. Second, ribosome. Yes, they have a ribosome. 70s ribosome of the in a prokaryote or in a monomers. Third, enzymes. Yes, they have enzymes, but we have studied that in monomers and bacteria, membrane-bound organelles are absent, and dear student, as you can see, this particular mitochondria is a double membrane bound. We will study in case of cell membrane, they are a double membrane bound organelle, and which are involved in ATP synthesis or providing energy in case of eukaryotic cell. So they are not present in prokaryote, so that becomes the correct answer. Next question. Methanogens are responsible for the production of, easiest question, they are involved in the production of methane, and here you are not seeing any particular option like the methane. Dear students, be remember that in biogas, if you talk, this biogas is made up of methane, CO2, H2, H2S, like this particular gases are found, and in this CH4 is a methane which is in 50 to 70% of total biogas. So biogas become the correct answer, which is involved where the methanogens are responsible for the production of methane. So let's talk about the next question, dear students. The future responsible for the survival of our bacteria in extreme condition is. So they have given the cell wall with peptidoglycan, and you know that archaebacteria do not have a peptidoglycan. Cell wall contains pseudoporin. Yes, it is possible. Cell membrane contain branched chain lipid. Yes, that is also possible. And production of methane. But most appropriate is which one? Cell membrane with the branched chain lipids that help to survive them in unfavorable conditions. Dear students, but NCRT what actually says? In NCRT they have given both cell wall and cell membrane are the responsible for this. And if the both one and two option is given, then which are the responsible for archaebacteria to survive in extreme condition? First option is given cell wall, second is cell membrane, and if fourth option is given both one and two, then you will attempt both one and two. Let's talk next question. The type of archaebacteria found in gut of ruminant animals like cows and buffalo, you know that they very well, they are the nothing but methanogens. Okay, so it is a direct question. We have just talked about the ruminococcus. Next question. The organism which can thrive well under extreme environmental conditions such as absence of oxygen, high salt concentration, and high temperature. And that is particularly, you know that they are nothing but archaebacteria. Next question. So I will be going in some fastly because these are the easiest question. Wherever the difficult question you will find, I will stop there and tell you, I will explain. The group of organisms that show most extensive metabolic diversity. It is the question which was earlier asked in Neet. Okay, so we remember these are nothing but bacteria. So 4 become the correct answer. Bacterial cell wall is made up of, easiest question, it is made up of peptidoglycan. The cell wall molecular bacteria are not referred as, it is referred as peptidoglycan, it is referred as murerin, it is referred as mucopeptide, but it is not a polyhydroxybutyrate. This polyhydroxybutyrate is a non-biodegradable component, so which is not found in the cell wall of bacteria, so it becomes the correct answer. Then the infolding of cell membrane, this we have just discussed this question, lysosome, repeatedly it is kept here. DNA referred as naked due to the absence of nuclear membrane, and the second option is not enveloped by the nuclear membrane. That is the correct answer for the bacteria to refer to DNA as a naked. Next, locomotory structure in bacteria are cilia, actually not found in case of the bacteria. Pseudopodia, actually found in case of the protists, not in bacteria. Flagella, yes, they have a flagella, and flagella is particularly divided into the basal body, hook, and filament. So flagella is the correct answer. Next to that, which of the following statement is correct? Yes, this is a good question. All bacteria are autotrophic. Dear students, we have studied the bacterial nutrition. Majority are heterotrophic and some are autotrophic, but all are not autotrophic. Next, all bacteria are heterotrophic. We can't say like that as well. Majority are heterotrophic. All bacteria are photosynthetic. It is not also possible. And some bacteria are autotrophic and some bacteria are heterotrophic. Then it is the correct one. And out of that, majority are heterotrophic. They depend on other organisms for their food. Next question is, which one of the following is said to be omnipresent organism? Omnipresent organisms are omnipresent means which are widespread, which are found everywhere, and you know that out of the given option, algae are not found everywhere. Bryophytes are also not found everywhere. Bryophytes are shady plants, they are found in shady conditions. Algae are found in aquatic conditions. Gymnosoma, the woody trees, they are found in some of the Himalaya regions and like this, but bacteria are found everywhere, so that becomes the correct answer. Next, asexual reproduction structure in bacteria is known as, dear students, a beautiful question. We have discussed, it is the easiest question, but it was asked in Neet 2 times. And that is known as endospore, unfavorable condition. Most chromatic organism showing oxygenic photosynthesis. It is a direct question. You know that cyanobacteria are the first organism to evolve oxygenic photosynthesis. So cyanobacteria, blue-green algae become the correct answer. Next, water plumes are found in polywater water body, often formed by, you know that water plumes are formed by the polluted water by the blue-green algae. So the green algae, brown algae cannot be that. Blue green algae will be the correct answer. That is a cyanobacteria. So 3 will be the correct answer. Apart from that, next move to the specialized cell found in blue-green algae meant for nitrogen fixation are, and you know that we have discussed this question earlier as well, it is heterocyst, specialized cell. Next to that, find the organism that not belong to the cyanobacteria. Chlorella, chlorella is a green algae. Nostoc is a cyanobacteria. Anabina is a cyanobacteria. Spirulina is a cyanobacteria. Now, dear students, here make one thing clear that spirulina is a cyanobacteria, but this spirulina is a cyanobacteria, but it is single-celled cyanobacteria and it is a protein rich. So we can call it as, and it can be used as a food for the space travelers. So we use spirulina as a single-cell protein. Okay, we use it as a single-cell protein, but it is a cyanobacteria. So chlorella is not found in case of cyanobacteria, so that becomes the correct answer because it is green algae. Next question. Hormone are found during asexual reproduction. Cyanobacteria are a type of fragments. Next, the red color of Red Sea is due to the presence of Trichodesmium erythraeum, Red Sea. So 3 becomes the correct answer. Cyanobacteria are chemoautotroph, chemoheterotroph, photoautotroph, and you know that they can synthesize their own food, so they are with the use of light, so they are photoautotrophs. So dear students, after discussing this many things, this many questions, and this many things in detail, I hope you all have understood very well bacteria. Now you can see bacteria in your all around, and you can visualize the characters of this bacteria. That will help you to understand this bacteria in detail and in proper manner. So thank you. Here we complete with this lecture. Here we complete this with this Monera, and in next class we will be starting with the kingdom